Welcome everybody to Unit 12, or excuse me, Chapter 12 review. So we're going to go over Chapter 12 review. Chapter 12 is nervous tissue, nervous system in general. Again, it's not a comprehensive review. You're responsible for all the material and the reading. As you go through the reading, if you have any questions, concerns, or comments, please email me and we can definitely straighten them out and get them figured out for you. So let's start with the review. Trigger zone. Basically, when it comes to a trigger zone, you should be familiar with all the parts of a neuron and what happens during the neuron's action potential. Trigger zone in particular is a question on there, so make sure you know what a trigger zone is. Action potential versus local potential, they're really kind of the same, right? Action potential means action down the line, right? So you're moving action potential in one direction. Local means what's happening in one particular area. Why is it a local potential? What is the result of a local potential, and how does it result maybe in an action potential? When it comes to action potential, you should understand the concept of all or none, right? It's either going to happen, it's going to fire, or it's not going to fire, meaning that you're going to create an action potential or you're not going to create an action potential. I may repeat this as we go through the review, but you should know everything there is to know about an action potential. What gates open, what gates close during depolarization versus repolarization. You should know what ions are being moved across the cell membrane, how many are being moved out, how many are being moved in, who are they, what do they do, and how does it result in action potential? How does it move in one direction? How do you get the action potential to go in one direction without it really coming backwards? Because as you know, action potential only goes in one direction. Some terms to be familiar with: summation, non-discremental, diverging, converging, ligand-related or ligand-regulated gates. There'll be some other terms like temporal summation you should be familiar with. You should be familiar with terms such as endoneurium, epineurium, parts of a neuron, how its structure is formed, how's it made up. Again, all the steps of an action potential. Again, like I said earlier, how do you produce an action potential? The key is really, how does it go in one direction? What does it mean to be all or none? What is that threshold that we have to reach? And in reaching that threshold, what happens during the depolarization phase? What happens during the repolarization phase? Right, as you see, I'm making that little, right, what you learned, that little the graph that you have in the notes. Again, neuron structure and all their functions. We look at neurons, we have the cell body, we have the soma, we have the Nissl bodies, we have axonal transport, we have different glial cells. So again, I'll mention those in a little bit, but be familiar with all those, as all the structures of a neuron and how they function, like the axon hillock, and so forth and so on. EPSPs versus IPSPs, be familiar with those neural integrations. What is an EPSP versus what is an IPSP? How do they work? How do they increase action potentials? How do they decrease action potentials, right? So if we have a IPSP and you're taking Valium, Valium's job is to slow things down. It slows your nervous reaction down a little bit, gives you that euphoric type feeling, decreases pain. Why? Because you're increasing IPSPs. That's what Valium and most of those oxycodone type medications do. They instill some type of activation of IPSPs in certain neurons. So neural integration is another term you should be familiar with. You should be familiar with another term called monoamine oxidase, right? It's an enzyme. What does it do? What does monoamine oxidase do? Nerve signals and the types of fibers. So we have those that are myelinated fibers versus unmyelinated fibers. We have thick fibers versus thin fibers. Knowing the type of fiber, if it's myelinated versus non-myelinated, how fast are we moving action potentials along a myelinated fiber versus a non or a unmyelinated fiber? What about a small fiber versus a large fiber in diameter? So don't forget, you have to think about resistance. The larger the diameter, less resistance. The small diameter, the more resistance. Of speed of action potential. Blood-brain barrier. What glial cells are part of the blood-brain barrier? How does it work? What's its job? So know the function of the blood-brain barrier and the cells involved. Back to glial cells. Well, astrocytes, obviously, right, are one of the glial cells that maybe be part of the blood-brain barrier, right? Don't forget about the satellite cells or the Schwann cells, the oligodendrocytes, the ependymal cells. So know those glial cells and their function. Know which ones, like the oligodendrocytes, they myelinate things. Do they myelinate PNS or CNS? Okay, so whose job does what, basically? Nervous system versus the endocrine system. These two systems go hand in hand. When it comes to the nervous system and what it does for our body, which is integration and communication, endocrine system is exactly the same way. It does integrate and does communicate. One does action potentials, the other one does hormones. So how do they relate? Which one's faster, which one's slower? Which one has a local effect versus a down-the-line effect? So know the difference, or at least in the chapter, within the chapter, how does it relate nervous system to endocrine system? When it comes to the concept of moving information from a outside the central nervous system to the nervous system, central nervous system, we have the afferent versus efferent versus interneurons, right? Afferent means toward the body, interneuron means inside somewhere, mostly likely in the spinal cord, which you'll learn in chapter 13. But then we have to have some efferent, or information leaving the spinal cord, or information leaving. So afferent in, efferent out, A towards, E away. Okay, so afferent toward the spinal cord, toward the interneurons, and efferent out. The more interneurons you have, the more information can be synthesized and could be readily available and used to create some type of efferent action. Most of our brain is interneurons, where we synthesize, where we categorize, we file information through these interneurons. When it comes to a simple reflex, it's afferent in, interneuron, efferent out. And that's something we'll go over when we talk about chapter 13. So know what an afferent versus efferent versus interneuron are, right? So for example, if we talk about effectors. Effectors is something that is affected by an efferent neuron. So a gland, for example, is an effector. Afferent in, interneuron, efferent out toward a gland. The gland is activated, and that activated gland becomes the effector because it was affected by an efferent input. When it comes to integration, like we talked about endocrine system versus nervous system, they both integrate and communicate. But the nervous system integrates information. The integration function of the nervous system is the function of the interneurons. It's the interneurons that allow you to integrate information. I'm going to go back and forth. So when it comes to, again, the action potential, be familiar with the refractory period, the relative refractory period. How do we prevent the action potential from going backwards? That's the key. CNS versus PNS, right? What structures make up the central nervous system versus those structures that make up the peripheral nervous system? What glial cells help in the central nervous system compared to those Schwann cells that work in the PNS, right? So we have the oligodendrocytes that will myelinate your CNS, and we have your Schwann cells that will myelinate the PNS. That's just one example, but know the structures and functions of the differences of the CNS versus the PNS. Threshold potential, let me go back before I go on. Threshold potential, another term you should be familiar with are post-tectonic potentiation, neuronal convergence. And I'll go over that in a second. Temporal summation, I said that already. So post-tectonic potentiation, you should know what that means. Memory and all the neurotransmitters involved. So memory, we have long-term versus short-term memory. We have have different neurotransmitters like dopamine, acetylcholine, substance P, nicotine, NMDA, and all the other fun stuff that make up those neurotransmitters that we use for memory. So be sure you know long-term versus short-term. And there you go, neurotransmitters and their functions. Neuronal regeneration, I think is a good explanation of that in the textbook. So be familiar with those different, basically how it works, how it happens. Visceral sensory and visceral motor versus somatic sensory and somatic motor. Know those definitions. What's a somatic sensory versus a visceral sensory? Obviously, visceral means organs. Somatic, we're talking about muscles. What's a visceral motor? Nerve. What's it do? Versus a somatic motor, which probably is going to move your muscles, right? Be familiar with different types of neurons, such as unipolar versus multipolar versus bipolar. Where are they located? Most of our multipolar are going to be up in our cortex in our brain, right? Because they have all those dendrites, and those dendrites are going to receive a lot of information, filter it down into one axon, which allows us to synthesize, integrate that information. We have a bipolar. Where are you going to find bipolar? What about the eyes? Check out the eyes and see what kind of receptors you have in your eyes. Unipolar, right? And most of our ganglia, most of our sensory ganglions, or next to your spinal cord, have that one little cell body, and out comes one, and then out it goes into a unipolar information. So be familiar with unipolar, multipolar, bipolar, and location of function. Also be familiar with axonal transport. There's different types. We got to get stuff. We have to get information, not information. We have to get material. What if that axonal knob at the end of your axon has an issue? What if it's damaged? We have to axonal transport proteins and structures and maybe some lysosomes, and maybe we got to make more things for that knob. Well, what's axonal transport? How does it get? What's fast versus slow axonal transport? And how does it get to that knob? Be familiar with that. Myelinated versus unmyelinated. Make sure you are familiar with those. Again, speed, where they're located. That's basically myelinated versus unmyelinated. Neurological disorders in the chapter. MS is in there. I think Alzheimer's in there. Going back to axonal transport, there's the fast anterograde versus slow anterograde. There's retrograde. So just be familiar with fast and slow and different grades, which way they go in antero or retro. Let me see. Cerebrospinal fluid, be familiar with that. Where is it located? How is it made? Who makes it? Which glial cell is responsible for making CSF? Multiple sclerosis, Parkinson's, Tay-Sachs, Alzheimer's, myasthenia gravis it's not on the test, but you should be familiar with those, whatever's in the chapter. Back to your ISPSs, we have the inhibitory local potentials versus excitatory local potentials. Be familiar with those. Again, we went over neurotransmitters, dopamine, serotonin, ACH, substance P. Synaptic vesicles is another term you should be familiar with. Calcium. How does calcium work? Calcium's pretty interesting, right? Without calcium, you can't release ACH from the synaptic knob or the axonal knob. Same thing. So in order for us to have a release of acetylcholine, I would have to have calcium influxing through the neurolemma at the axonal knob in order for us to get those vesicles to migrate toward the end, meld or mold with the neurolemma, open up the vesicle to release ACH into the synaptic cleft so it could go off to its receptors. So we need calcium. Too much calcium in our diet or too much calcium in our blood could cause an overactivation of our nervous system. Not enough calcium in our blood could cause a decrease in activation or more of a relaxing type issue with our nerves, causing paralysis, flaccidity, not necessarily paralysis, but could turn off your muscles, right? So calcium, what's its function? How does it work? Pretty much told you just now, and see how it relates to that information. So that's about it. Any questions, concerns, feel free to email me. And it's a big chapter, but you'll do just fine. And good luck.